Hello, and welcome to the latest of the Kavli Foundation Spotlight Live webcasts, which offer a chance to hear from scientists on the cutting edge of discovery. In the next 30 minutes, with the help of two astrobiologists, we'll travel to some of the most extreme places on Earth and in our solar system in search of microbes. One of our guides will be Jocelyn DiRuggiero, a microbiologist at Johns Hopkins University, who is currently studying how microorganisms survive in the driest place on Earth. The other is Chris McKay, a senior scientist at NASA Ames Research Center, who studies the microbes that live in Mars-like environments here on Earth and helps NASA plan missions in search of life. My name is Lindsay Borthwick, and I'll be moderating today's hangout. As Chris and Jocelyn can attest, the Earth is home to some pretty awesome and unusual microorganisms. There are bacteria that can eat radioactive metals, others that can tolerate lethal doses of radiation, and still others that persist in the frigid waters beneath the polar ice sheets. These organisms are fascinating in their own right, but it's what they are teaching us about how to hunt for life in the cosmos that we'll be talking about today. The search for signs of life elsewhere in our solar system is imminent. Missions are being planned to Mars in 2020 and could be followed by probes to Saturn's icy moon, Enceladus. Astrobiologists hope that these missions will yield conclusive evidence of life, but to get there, they first have to decide precisely where to look, what signs of life to look for, and how to detect those signals. That's where microbes called extremophiles come in. They, along with the harsh environments they inhabit, serve as living laboratories here on Earth for what scientists hope to discover beyond it. Chris and Jocelyn, thank you for joining me.
Welcome.
Yeah, good to be here.
Before we get started, I'd like to remind you that if you have a question you'd like to ask, please send it by email to info@kavlifoundation.org or via Twitter using the hashtag KavliLive. All right, so we'll get started. To give our viewers a sense of where your science has taken you, I'm wondering, what are the most extreme places on Earth that you've visited, and what did you find there, microbially speaking? Jocelyn, do you want to start?
Yeah, sure. For me, it would be the Atacama Desert, which is in northern Chile, and it's one of the driest and oldest deserts on Earth. I mean, there are places in the desert where it hasn't rained in more than 50 years, so it's very, very dry. And there we found flourishing microbial community inside rocks. Those rocks are translucent, so they can carry out photosynthesis, and this is the best place they can find refuge, where they're protected against the elements and where water can be retained longer. And so there are places like rock made out of sodium chloride, I mean, you table salt, and inside of those rocks you can find those communities. And very many different other substrates. So to me, it's totally fascinating. I mean, the etching life inside rocks. How can it be more extreme than that?
That does sound pretty extreme. What about you, Chris?
Well, the Atacama Desert, as Jocelyn just indicated, is one of my favorite places, too, for just the reasons she said. The other place that comes to my mind is the coldest desert on Earth. The Atacama is the driest. The coldest is in Antarctica, an area called the Dry Valleys. And there we also find microbial life hidden in obscure places. We find it under ice, and we also find it in rocks, similar to what we find in the Atacama. So it seems that living in rocks is a good strategy for microbial life in these extreme Mars-like environments.
Okay, so what are the new frontiers for research on extremophiles? Jocelyn, in our roundtable discussion, which we've published online at thekavlifoundation.org, you mentioned one of the new frontiers is extremely low nutrient environment. So you're looking for microbes that survive with very little, with very few nutrients in their environment. But can you tell me about that one or others?
Yeah, so this is a good one in the sense that the surface of the Earth we use to what's called primary production via photosynthesis. So microorganisms make organic carbon using energy from light, and you have this whole food chain. But in places like subarctic lakes that are covered all the time by a very big sheet of ice, you don't have light. So microorganisms have found other way to build this organic carbon to then be able to feed the rest of the community. And so that's very interesting, and that's very applicable to extraterrestrial environment. You know, if you're talking about the ocean of Europa, it's not going to be like down there. If you're talking about the subsurface of Mars, it's also true. And so looking at those places on Earth can teach us what type of strategies microorganisms can use. And what I mentioned before that is very interesting, for example, is these microorganisms that grow very deep in a gold mine in South Africa and derives this nutrient from the radioactive decay of uranium. So here you have radioactivity that somehow end up producing the nutrient for this organism. So I think those are very interesting type of environment. And of course, the dry limit for life. I mean, we don't really know what that is, but this is becoming very relevant, not only for the exploration of our solar system, but also in terms of exoplanets. You know, how far, how close to a star can a planet be, how dry can it be to support life, and how can we choose targets based on that. So those are really two areas that I think are progressing and very interesting right now.
Yeah, I can add another area that's related to what Jocelyn was just saying, and that's the response of these microorganisms in these slow-growing, dry environments, the response to external hazards like radiation and oxidants, how they deal with this. When organisms are growing fast, like us, it's pretty easy for us to repair the damage that's done by background radiation and environmental oxidants. But when you're growing so slowly that it takes you thousands of years to reproduce, these small cuts could accumulate and could be a real problem. And how organisms deal with that is something we're just beginning to understand on Earth, and has a lot of implications for how life might survive or have survived on Mars or in the outer solar system where radiation levels can be quite high.
All right. Jocelyn, you mentioned these analog environments, so these places on Earth that we look at because they resemble, or there's a close a proxy as we can get to places in the solar system. We have a question from a reader who wants to know, what kind of microbes would you expect to find in a place like Enceladus, where temperatures reach, you know, around about minus 200 degrees Celsius, and which Earth analogs or extreme environments could aid us in figuring that out?
So we would find microorganisms that somehow can resist very cold temperature, but probably also salt, high salt. Because if you want to have liquid water at such low temperature, you're going to have to increase the salt concentration in the water. So good analogs for this type of environments are lakes in Antarctica, those subarctic lakes that are covered with a very thick layer of ice. And so those are good analogs in terms of temperature, as I mentioned also earlier, in terms of the nutrients that those microorganisms could use. So how do they make a living? And so those are very good questions that we can answer by exploring those lakes. So Lake Vida is a good example, for example, that has been explored right now. Lake Vostok, although we're not sure what's happening with that. So yeah, subarctic lakes would definitely be a good place, a good analog environment.
Okay.
Yeah.
Let me add a little to that. One of the reasons we think, one of the ways we think Enceladus maintains the liquid underneath the ice, the ocean and below the ice, is through a process called serpentinization, which is a reaction of water with the iron in the rock in the core of Enceladus. And that reaction releases heat, but it also releases hydrogen. And so organisms can use that as an energy source. As Jocelyn said, sunlight doesn't reach down to the ocean on Enceladus, but hydrogen is a very useful energy source. And there are subsurface ecosystems on Earth which also rely on hydrogen released from water-rock reactions as their energy source.
Huh. So there really are surprising similarities.
Yeah, there are definitely deep sea vents that are based on serpentinizations. And so those are actually quite well-studied systems. And the organisms oxidize the oxygen, and that's how they generate the energy they need to be able to fix carbon and then function and grow. Yeah.
Okay, Chris, when you're planning a mission, and this is a big question, but how do you factor in this kind of research on microbes and the analog sites in which we study them here on Earth? What kind of decisions does it help you make?
Well, I think the important way that this kind of research informs our planning for missions is deciding what kind of instruments to send on a mission and what to measure. If we fly, for example, through the plume of Enceladus, we know it has organic material. We know it's coming from a water environment. What should we measure? Should we look for amino acids? Should we look for lipids? Should we look for photosynthetic organisms? Obviously not. So these analogs provide concrete data on what kind of signatures life in these environments might have and what kind of instruments we can build. And then we can take these instruments and test them in these environments. Obviously, if our instruments can't detect life coming out of a deep sea vent or in the Atacama Desert, then they're not ready to go to Enceladus or Mars. So the testing on Earth is very important, and these environments, extreme environments, provide a good test bed for us.
Okay. Have we got any of those instruments that are ready to go?
Well, we've got some instruments that we would propose right now, for example, mass spectrometers. But the one that I'm the most excited about and pushing right now is instruments that can detect amino acids. I think amino acids may be the best clue for searching for evidence of life on Mars or in Enceladus, because life uses amino acids. It uses only certain amino acids, and life elsewhere probably needs amino acids also if it lives in water, but it might use different ones than life on Earth uses, so it's got some interesting possibilities for detecting not just life like Earth, but life that's a little bit different from life on Earth. So that's where we're going next, I think, amino acids.
All right.
Right, and of course this is within the solar system, right? And then you have this whole field of exoplanet outside the solar system in places where we can't really go right now. So then we can't really fly an instrument there, and the best we can do is use spectroscopy to analyze the atmosphere of those planets, and then being able to figure out what can be a signature for life is another big issue that we need to address.
Okay. And what kind of signatures can you look for on exoplanets that might signal that they're worth further exploration?
Well, that's the big question, right now. So, I mean, you could think about oxygen because life on Earth is based on oxygens. But, you know, if you look at Earth history, oxygen has only been around for maybe 1.2 billion years. So the question is, you know, what else do we measure? And also the idea that some of those gases that we're going to measure in the atmosphere might be produced by processes that are not from life. So actually what people are thinking right now is that if you have a disequilibrium, which means if you have two gases that would normally not exist together because if they do, they interact with each other and make something else. So if those two gases persist side by side, it means that one of them might be produced continuously, and that might be a sign of life. So I think that's the big idea right now. It could be oxygen and methane, you know, CO2 and oxygen and other things like this. So the equilibrium of the gases.
Let me just add that the oxygen is particularly interesting. As Jocelyn said, it's used by life, but it's also, at least on Earth, so abundant that it would be easy to see. So in terms of what might be the next breakthrough in terms of searching for life on exoplanets, I'd be willing to predict that it'll be at first a detection of oxygen. Then that will be followed up, as Jocelyn indicated, by a search for other gases, which make the story even stronger if they're found as well. So, for example, on Earth there's oxygen, but there's also methane in the atmosphere, much lower levels.
Okay, I have another viewer question, and it's for you, Jocelyn. Would the life forms embedded in the Atacama rocks that you study survive a trip from Earth to Mars as a fragment of an asteroid impact?
Well, that we don't really know. And so we're actually planning some experiments where we want to take some of those rocks and place them in what we call a Mars chamber, which is a chamber where we could apply a different type of environment, conditions very similar to Mars. So in terms of temperature, but also pressure, which is in space an issue, and also different level of moisture in the air. and see how well those organisms could survive. I mean, there are definitely organisms that we have found on Earth that have survived for thousands, even millions of years inside, for example, salt rocks. And then people have been able to extract the DNA, they've been able to make them grow again. So we know it's possible, but the issue in space is the amount of radiation that those organisms are going to receive and how they're going to repair when they get to their destinations, and is that possible? So those are also very important issues that we have to look at.
Okay. Now, recent discoveries have raised the possibility that life on Mars may still exist, and I'm thinking of the Curiosity rover's detection of methane near Gale Crater, but there's also been laboratory work done on how bacteria grow under Martian conditions. Chris, are you more optimistic about Mars than ever?
Well, I'd say yes, we're optimistic. We're optimistic that we may find traces of life in the past. If there's life there now, it's probably below ground. There's optimism growing on that possibility, but it's also a little bit daunting because the ability to reach it and investigate it is hard. Drilling on Mars is a challenge that we still don't really have a good handle on.
Okay. Now, you've said that Titan, which is one of Saturn's moons, is another candidate for life in our solar system, yet it has no water. And life, at least life that we know it, requires water. So what makes you think that Titan's seas, which are filled with liquid hydrocarbons like methane, could support life?
Well, to me, the interesting thing about Titan is it's the only world besides the Earth that has beaches. And what do I mean by that? A beach is where a liquid comes onto land. Right. That interface between liquid, land, underneath the sky is on Earth, and the only other place we have it is Titan. The difference, of course, is the liquid on Titan is not water. But it challenges our concept that life requires water. Maybe life just requires a liquid. On the other hand, when we look at organic chemistry, the important role that water plays, it's hard to fathom how a liquid besides water would do the job. But that could just be our own parochial view. We like water because we were born in it. Organisms on Titan may be looking at Earth and saying, well, there's a liquid there, but who would ever want to live in water? Right? So it may be a question of perspective, and we're just beginning to investigate, could the beaches on Titan have life forms that live at the interface between the liquid and the land? I think it's an interesting question, and nature, in a sense, has given us this challenge, this opportunity, and we want to investigate.
Interesting. Okay. Now, one step in the evolution of life on Earth is the tendency to form co-ops. So microbes very often come together in complex cooperative systems, such as microbial mats in hot springs or even microbial biofilms in the human gut. Jocelyn, why do they do this, and could this tendency to form co-ops help us search for life elsewhere?
Well, I think microorganisms are very much like any organism on Earth. They have to live in community to be able to survive and reproduce. And the community works as a system where different part of the community might have different roles. So, for example, some of the organisms can produce the organic carbon, others can fix the nitrogen, and then they can also exchange genetic material. And that allows them to be more responsive to changes in the environment. So if you think in terms of antibiotics, for example, those are typically resistance to antibiotics can be laterally transferred between microorganisms. They can exchange genetic material to become resistant. So if in your gut you have microorganisms that are and that aren't, they're different. But if they live together, they can exchange this resistance, and then the community becomes resistant. So this is really part of how any living system works on Earth, and it's not just microorganisms. So biofilms are the same, and microorganisms living in the guts of humans and animals are the same. So how is that going to help us with finding life on other planets? Well, this is just the way they are. We have to look at communities. And past is the time where we're just going to look at how one type of microorganism survive in extraterrestrial conditions. This is not what we want to do. We want to look at communities as a whole on extreme environment in Earth and see how far we can take those communities, how far we can stress them and push them and evolve them.
I see. Okay. I just hosted another live webcast on the relationship between the human gut microbiome and the brain, which viewers can find online. So I can't help but wonder if all of this interest and experimentation and tool development that's happening around the human microbiome is spilling over and affecting the work that you do.
Oh yeah, absolutely. I mean, developments of tools, high throughput, how you can analyze a very large number of samples at once, and then also bioinformatic tools to be able to analyze the very large amount of data we get when we do that type of molecular work. So in both cases, we're looking at communities. We're not trying to isolate the microorganisms. We're just extracting the genetic material from the whole communities, trying to get the sequence of all those genomes, of all the genes, and then you just have to apply computer tools, computational tools to be able to figure it out who is there and what they might be doing. So everything that is being developed, with the human microbiome can be applied to any type of community. And it's actually very funny because I'm working in rocks in the Atacama, but I'm right now writing a proposal to look at the gut microbiome in mice. So, it's just a different type of community. It's not, an environment as exciting as the Atacama, the mountain ability, but at the same, the same ecological principle of the functioning of a community and the same tools to get at how we're going to decipher this functioning.
That's interesting. Chris, I guess we're used to technology transfer happening usually from space agencies and the military into the public, but is the opposite happening here? Are you able to?
Definitely. In the biotech area, we are the beneficiaries of this enormous technology being developed for genome sequencing and so-called omics. So there's an enormous technology step that's taken over the last 20 years in our ability to analyze and characterize these microbial ecosystems, driven by interest in things like the human gut biome and gene and disease and so on, and that provides a technology base for the next generation of life detection tools. So we are getting, you could call them spin-ons. They're not coming from NASA. We're benefiting from the research that people like Jocelyn and others are doing in areas that are deal with mice intestines and cows and chickens and disease and all those sorts of things.
Okay. Now, President Obama mentioned in his State of the Union address last week that NASA astronaut Scott Kelly is heading to the International Space Station for a year, and that scientists will study how that prolonged journey affects his health, including his microbiome. I know this isn't your direct area of expertise, but I'm wondering, why is his microbiome of interest? And do we understand what happens to life or to microbes when gravity changes?
I can take these questions. I think his microbiome is essential. With our current research, we are finding that there is this gut-brain axis where you have these direct interactions between our brain and the hormones that we produce and the gut microorganisms. Those astronauts are going to be put in an environment where the circadian rhythm is going to be disrupted, this 24 clock that we have. The feeding habits are going to be disrupted. This is going to have a direct impact on the microorganisms in the guts, and as a consequence, how they react with the immune system, how they process some of the hormones that we're producing, and that's going to have a feedback on the cognitive ability, how they're able to function in space, what they can do, how well they can think, how they can sleep. So I think this is essential, and this is really research that is being done right now at the moment. And I was mentioning the project I'm working on with the mice. We're actually going to be disrupting the circadian rhythm of these mice. We're going to put them in jet lag, and we're going to see exactly what happened in the gut and how this is impacting their performance, but also if they get depressed, if they have mood dysfunctions, and this is directly applicable to what happens to the astronaut in space or anybody doing shift work or jet lag. Definitely, yeah.
Okay. Now, some people have put forward the idea that life on Mars may have seeded life on Earth. But do you worry about microbes hitching a ride the other way, Chris?
Oh, indeed. Indeed. We have international rules that dictate the cleanliness levels on spacecraft for just that reason. We're very concerned about carrying life from Earth to Mars, both because it will contaminate our scientific experiments, but also because we could inadvertently put life there and then create a problem, an ecological problem on Mars. So we do have to worry about that. And people say, well, isn't it possible that life on Earth and Mars are naturally exchanged? And yes, that's a hypothesis. But still, it's prudent to clean our spacecraft and do clean technique and not carry stuff with us that we don't know what the effects would be. So even accepting the fact that it's possible that life on Earth came from Mars and life on Mars comes from Earth, but there's exchange, we still need to be clean in our approach to investigating the life on Mars.
And do those rules apply also to the commercial spaceflight industry?
They will. They are set by international treaty, so any U.S. company bound by U.S. law, international treaties to which the U.S. is a signatory, becomes national federal law, and so any companies operating in the U.S. that want to go to Mars will have to abide by those rules. And I believe the same is true for companies operating in any other country. So they are somewhat similar to laws regarding the pollution at sea and so on, and they will extend even to private operators when they get involved. And there's really not much pushback against these rules. They make sense. There's no point in going to Mars and releasing things in the environment that are that you don't understand or that are dangerous. And this doesn't rule out human exploration. It just means that when you do send humans, you have to be careful how you do it and what you do.
I see. Okay.
And it's also the opposite. I mean, planetary protection for bringing samples back to Mars. And there's a lot of rules that have to be applied before we can bring any samples back from any other environment. So it goes both ways.
I see. Okay. We're just about out of time, but Chris, I was wondering if you can give us a taste of what is around the corner in terms of searching for biology in our solar system. I mentioned Mars 2020, but are there other very likely missions you can tell us about?
I think there's 3 targets that are in our sights: Mars 2020 and other missions. European mission, ExoMars, will be very exciting. Then Europa. Europa is there's a groundswell of enthusiasm for doing a mission to Europa. It's very challenging because of the radiation and the thick ice, but there's a push to do it. And then Enceladus, with that plume out there in space, it's almost like there's a big sign: free samples, organic rich, take one. So I think those 3 worlds are going to be the target of a series of astrobiology missions. They're going to be very exciting in the next 10 years.
Yeah, that's right. And we had this question last time, and we also need to add to that the possibility of discovering life or evidence of life on exoplanets. And so Chris had decided this is an arm race between the astronomers and more the planetary scientists.
And synthetic biologists.
Right.
Yeah.
Exactly.
Okay, so then just the last question is, do you think that the question of life elsewhere will be answered in our lifetime?
Oh gosh, I hope so.
Stop smoking, exercise regularly, and extend your lifetime.
All right. Well, this has been fantastic. I've learned a lot, and I'm sure our viewers have too. Thank you both for participating today, and have a great afternoon.
Thank you, Lindsay.
Thanks.
Thank you.